Hey, this is Mr. Peterson. I'm going to try to go through cellular respiration in 300 seconds or less. So kind of a challenge. So we'll see if we can do it in 5 minutes. Keep in mind that there's also 3 other recordings that break each of these reactions down a little more elaborately. But this is I thought, let's try to do this in 5 minutes. Now, the clock hasn't started yet, but it will. And now it's on. Whew. So cells break energy and they use it to make ATP. So coupler, the ATP is the coupler. So here's that big formula. You got to know this. Glucose plus oxygen gives you carbon dioxide and water. So what we're going to do is we're going to follow that glucose molecule and particularly concentrate on the carbons and the hydrogens and what happens. So here's a cell, nucleus, mitochondria. Cells have lots and lots of mitochondria depending on what type of cell it is. So lots of mitochondria. We're going to zoom in on one. So here's one mitochondria. So the mitochondria has some anatomy that we have to pay attention to. It has a couple of membranes. It has the space in the middle, which is called the matrix. It has an inner membrane space, which is the space between the outer membrane and the inner membrane. And what's really going to be important is that inner mitochondrial membrane, down to 4 minutes. So here's a sugar molecule. 6 carbons. So pay attention to those 6 carbons. We're going to follow those through a couple of trips through cellular respiration. How does it get into the cell? Facilitated diffusion. First thing that happens is that molecule is broken apart. So one 6-carbon molecule is broken into 2 3-carbon molecules. Those are called pyruvates. And that happens in the process of glycolysis. That happens in the cytoplasm of a cell. So a couple of 6-carbon molecules. Now, eventually what's going to happen is that pyruvate is going to be broken down some more, and we're going to lose the first CO2. So that's a carbon dioxide that we now exhale. So this 2-carbon molecule heads into the inside the mitochondria, into the matrix, and what's going to happen? We're going to follow it a little bit closer. Oh, hey, we got a new 5 minutes. I like that. We'll just keep going. So that 2-carbon molecule takes part in a series of chemical reactions in the matrix. It's called the Krebs cycle. And what happens is the Krebs cycle takes that 2-carbon molecule through a series of chemical reactions. Eventually, all of the carbons are split apart. We exhale those as carbon dioxide. And it captures the hydrogens and the electrons from the glucose. So that H12, we're after those hydrogens and those electrons. So remember, this happens twice because we had 2 3-carbon molecules to begin with, so the same thing happens. And eventually we get to having a molecule called NAD. NAD is an energy shuttle. Oh, hey, we got another 5 minutes. I like that. It just keeps going. So the NAD collects the hydrogens and the electrons from the glucose molecule, and it's going to act as a shuttle to the electron transport chain. So we've had glycolysis, happens in the cytoplasm, the Krebs cycle, which breaks apart carbon molecules and puts them into the matrix of the mitochondria, and then we're going to look at that inner membrane of the mitochondria, and that's where the electron transport system, the electron transport chain, is going to build a whole bunch of ATP with those hydrogens and those electrons. So here's a look at that membrane. So we've got a bunch of proteins that are embedded in this membrane, and they're going to be important in shuttling electrons through. We're going to use the energy from electrons to drive some hydrogens from NAD over to that space, the inner membrane space. So that's going to happen a bunch of times. So that electrons are going to be used to power those protein pumps to put a whole bunch of hydrogens. So we're going to make a proton gradient, a hydrogen gradient on one side of the membrane. So all kinds of hydrogens are going to pile up. So it's kind of like water behind a dam. So we've got hydrogens that are going to pile up on one side of a membrane. You can hear my dog barking in the background. I'll have to scold him later. And those hydrogens are going to be used to make ATP. So that brown protein is going to build some ATP. So the oxygen, which you and I take in, is going to bond with those hydrogens. So the oxygen actually winds up to be the final acceptor for all of those hydrogens from our food. And those hydrogens, as they go through that protein, are going to be used to take ADP and a phosphate and build ATP. So this is the big machine that builds most of the ATP. So if you remember, glycolysis only produces a couple, whereas electron transport chain is going to produce 30-something ATP for every sugar molecule. So that ATP is the coupler of all of our chemical reactions, exergonic and intergonic reactions. So the hydrogens, which are going to get used up to build lots and lots of ATP. And that ATP is going to be recycled into ADP, and we're just going to use it over and over again. It's going to be sent back into the mitochondria to get recharged. Cellular respiration. I know that was probably more than 5 minutes, but it was pretty quick. So I hope that helps.